You've just finished running 200 protein variants through a week-long workflow. You've done expression, purification, and run multiple assays on your variants. Then someone asks, which plate did sample 147 come from originally? And you cannot answer. When you're running hundreds of variants through so many steps, there's so much information that without proper sample tracking, you can't see the forest for the trees. One plate swap wasn't tracked correctly, and now you need to trace it back all manually. In high throughput engineering, for every protein, there are many steps in the workflow. Every characteristic you measure, from DNA sequence to binding affinity to thermostability, needs to be tied together. Sample tracking ensures exactly that. Without proper sample tracking, ML models miss crucial context. We need every measurement linked to the right protein sequence, because that's what makes high-quality training data. Today I'll show you how we build a flexible sample tracking system that maintains complete traceability while being flexible enough to adapt to our constantly evolving workflows. Hi, I'm Daan, Research Associate at Cradle. At Cradle we help scientists engineer better proteins faster using AI. But here's the challenge. Our ML researchers need as much context as possible about every protein variant they work with. When assay protocols change or new instruments come online, how do we ensure data integrity doesn't break? The answer is robust sample tracking. It's the hidden foundation that makes high throughput experimentation possible. Sample tracking seems simple until you scale up. In our Amsterdam lab, we process hundreds of samples across different stages, from DNA assembly and sequencing through protein expression and normalization to our final measurements. At every stage, information is added to the sample. Which DNA sequence, what expression condition, which batch, what assay results. Tracking samples allows us to be confident in our data, troubleshoot issues, and reuse our data more easily. We've built our solution on Benchling LIMS, using a nested sample structure that follows our protein engineering workflow. It starts with protein designs from our ML models. These become DNA parts, which gets assembled into constructs. These constructs get sequenced and then become samples that produce proteins. Every sample links to its parent sample and creates a complete family tree. This means we can trace any measurement back to the original protein design. And the most important part, it's flexible. You can just add or remove any step without breaking the system. This really matters because we are always looking for ways to improve our workflow, because it's always in motion. Let me walk you through how this works in practice. Starting with protein designs, we generate corresponding DNA sequences. And after synthesis and assembly, our in silico workflow processes sequencing data and creates several entity types in Benchling. The magic happens with our hierarchical structure. Each sample has calculated fields that automatically pull information from its parent samples. When we transfer these samples to new plates or run a QC analysis, new child samples are created with complete inheritance of the metadata. This creates flexibility. We can adapt protocols or add new assays without breaking our tracking system. Here's how it works for a specific sample. The DNA part gets combined with a backbone and becomes a vector with associated sample. As this sample moves through expression, normalization, and the final assays, every step creates a child sample that inherits the complete history. A researcher or scientist can instantly trace any result back through the entire workflow. This system delivers 4 critical advantages. Complete traceability. Every data point links back to its original design. Workflow flexibility. We can modify protocols without losing tracking. Automated metadata inheritance. No manual data entry means fewer errors. ML-ready data. Our models get the full context they need for training. We're continuously enhancing this system. Next priorities include better variant lookup tools and direct instrument integration to eliminate manual data transfer entirely. Sample tracking might be invisible when it works, but it's the foundation that enables everything else. Reliable data, trustworthy ML models, and ultimately better proteins. The faster and more reliable we can track samples, the faster our AI can learn and improve. In ML-driven protein engineering, data quality is everything. If you like this video, make sure to follow and subscribe to get more of our lab protocols and workflows. Until then, thanks for watching.